Kia ora and welcome to the June LINKOnline Webinar. We're really sorry that you can't see us online again today. It's a technical glitch with the GoToWebinar platform. But I want to assure you that we're all here and we're all looking very smart and smiling. My name's Christine Harper and I'm the Stakeholder Relationship Manager at Manaaki Whenua. I love working alongside our scientists to share their research, and we hope this will inspire you to think about how what we're learning can be useful for your own work. This is the first in a mini-series of webinars to share information from Kiwi Rescue, a 5-year research program to help reverse the decline of kiwi. This program is led by Manaaki Whenua and closely aligns to programs at the Department of Conservation and with other partners around the country. And it's already delivering new knowledge to help conservation efforts. 2 further webinars will follow in July. But today we're hearing from Dean Anderson. Dean is a research scientist specialising in spatial modelling of disease and pest vertebrates in New Zealand. His work helps understand the spatial distribution of birds and other species to be protected and the pests that prey on them. A fascinating talk awaits. I'll be back towards the end to help with the Q&A session. So do get your questions into the chat box in plenty of time. We might not get through all questions in our live session, but rest assured we will follow up on them all and publish responses alongside the webinar recording on our website. Over to you now, Dean.
Thank you, Christine. Kia ora koutou. I'd like to begin with acknowledging my collaborators on this project and a research grant, which Christine has just been talking about, from MB. So in recent years and decades, extensive field work has really increased our ecological understanding of native fauna and introduced predators. And we've become reasonably good at actually controlling invasive predators in defined areas. And I think we would all agree that scaling up that management of invasive predators to very large area would really improve our impact or our outcomes for biodiversity and allow some species a fighting chance of persistence in mainland landscapes well into the future. So scale in this case, or broad scale, is very flexible. If you look at Eglinton, shown up here, the Eglinton management area shown in blue with the trapping line in red, this is a relatively large area. But for our purposes today, when we're talking about scaling up, we're scaling up to all of Fiordland, or it could be half of Fiordland, but in our case, we're talking about all of Fiordland. And a main, and so in this project, we are really trying to take what has been learned in ecological studies and through management experience and put that together in a model that we can forecast ecological outcomes given different management strategies over very, very large areas. And a primary motivating factor for this work is developing a mechanism or a methodology for aligning management objectives with what is actually feasible. Obviously, applying management over very large areas is costly. And so given limited financial resources, what is actually feasible that we could hope to achieve? And so we advocate that any project, broad scale or fine scale, really should begin with an exploration of the ecosystem at hand, and with a model such as this one to identify what are feasible and realistic outcomes. And those will help align expectations of policymakers, managers, the public, and research scientists. So, sorry, one too many. The system or the case study we're talking about today involves forest productivity, rats, stoats, and kiwi. And it starts with the underlying forest productivity and masting events. And a mast is when most trees in a very large area all produce fruits or flowers, fruits, and seeds about the same time. And this produces a huge resource pulse, which rats benefit from. I'm sorry, I've jumped ahead here. Okay, so rats increase due to that resource pulse. And that feeds the stoat population, which then depredate kiwi, and the kiwi suffer as a result. But 1080 can interrupt this cycle. Rats will eat 1080 baits and die, and stoats don't eat 1080 excuse me, baits, but they eat the rats that do eat 1080. And therefore, rats really act as a vector for getting 1080 into stoats, which reduces the stoat population, and kiwi can then benefit and thrive. Now, 1080 is really the only current available tool that we have to control predators over very large areas. And so that's our primary focus in this modeling. We do include trapping, but 1080 is the primary management tool to achieve broad-scale outcomes. So how do we do this? If you can imagine this is a beginning of a conceptual description of our modeling. And over here on the left, we have imagine that's a big landscape, and we've broken it down into grid cells to help illustrate the scale issues involved here. And at fine scale, as shown in this black square here, we model those dynamics that I just described about forest productivity, rats, stoats, and kiwi. And so that involves modeling survival, reproduction, competition, and predation, which all play out in very localized areas. At broad scales, we have forest productivity and masting events, which occur periodically over time, but the spatial distribution and arrangement of those masting events will vary somewhat unpredictably. And disturbance via 1080 is the next broad scale process or event that occurs. And while we could include extreme weather events, our primary disturbance event here is 1080, which selectively kills rats and stoats in that area. Now, note that the 1080 distribution or application covers a very large area, but really in this model, it only covers a fraction of the landscape. So we really are scaling up to very large areas. And then the last broad scale process that we're dealing with is dispersal. And that's the movement of individuals, rats, stoats, and kiwi from one area to another. And so if you imagine this red area where stoat population would be knocked down by 1080, that stoat population won't remain low forever, because survivors in this area will reproduce, but also individuals from outside that area can recolonize it and give a boost to this population. So these three broad scale processes, they really act to synchronize or link very widely separated dynamics going on at fine scales. So the primary question we're addressing today is, how much will a kiwi population grow or decrease given a predator management strategy over a 20-year period? And we're talking about broad scale here, so we're necessarily interested in a broad temporal spatial scale as well, because what happens over the next year or 2 years probably isn't important. But what we're really interested in is getting outcomes over an an extended period. I just want to back up to here and talk about these polygons here, these colored areas. These are management zones in Fiordland. And these are the areas where 1080 can be applied. And these black areas are just trapping networks. So this is the Murchison Mountains for Takahe, Secretary Island, and Fiordland, Resolution Island. And so we can address the question of how kiwi fare over the entirety of Fiordland or within these individual management zones over time. And so while we explore a wide range of management strategies, a principal grouping factor in across our strategies is how we apply 1080. It can be prescriptive, so that 1080 is rotated across all the management zones every 3 to 4 years, or it can be reactive. So for example, if we're monitoring rats with tracking tunnels, as shown over here, these are tunnels that are deployed across the landscape, and we can see rat tracks going through it. If 20% of these rat tracking tunnels detect rats, then that could trigger a 1080 operation, because that would show that the rat numbers are increasing, and we would expect stoat numbers to increase in turn. Or another reactive strategy is to respond to masting events. If, for example, 50% of a given management zone is in mast, then that might trigger a 1080 operation in that particular management zone. So if we jump to some results and we look at projected populations for rats, stoats, and kiwi, if you take this vertical dotted line as today, this is time 0 in our modeling, and we model management scenarios going forward in time 20 years. And each of these lines is a different management scenario. So this is a subset of scenarios that we've considered for illustration. And a lot of these scenarios for rats, you'll see, do not decrease the rat population substantially. Whereas these 3, they do decrease the rat population. And similarly with stoats, several of the scenarios don't decrease the stoat population substantially over time, but these 3 result in reduced and populations that are maintained at low levels. In contrast, these 3 scenarios do very well for kiwi, or kiwi do very well under those management scenarios. There's a lot of scenarios where kiwi actually decrease, continue to decrease, or increase only slightly. So this slide shows how kiwi outcomes vary with the number of 1080 operations. Now, if we applied 1080 everywhere all the time, we would probably have really good outcomes for kiwi. But while we're not modeling cost explicitly in this talk or this at the moment, 1080 operations are very expensive. And so we want to minimize the number of 1080 operations. So on this down here on the x-axis, we want to minimize the number of 1080 control operations, while we want to maximize the annual kiwi change over 20 years. So where we really want to be is up in this corner with a few operations, and the kiwi do very well. Well, we're not there yet, but you can see the individual scenarios and their identifying markers. So these numbers just indicate, or just an identifier for a scenario. So scenarios 12 and 13 in this graph are the best scenarios, because they minimize the number of operations and they maximize the annual kiwi change. And these scenarios are reactive to rats. So they entail monitoring rats with tracking tunnels, and then only when the rat tracking tunnel rate is above a given threshold, 1080 is applied. Now, scenario 11 does very well for Kiwi as well, but you'll see that it requires doing about 60 1080 operations across Fiordland over a 20-year period, which is a lot of 1080 operations and very, very expensive. And this is a prescribed strategy in which 1080 is applied on a 3-year rotational basis. So the best models are reactive, require monitoring rats, and are reactive to elevated rat abundance, such as when tracking tunnel rates are greater than 20%. Interestingly, responding to masting alone was not effective. And what's going on here is that a rat population can be at relatively low, quite low densities going into a mast. And if that's the case, then the rat population may not grow to really high levels despite the huge resource pulse created by the masting event. And in that case, it may not be effective to deploy 1080 in those areas where a masting has occurred and resources have increased, but the rodent population has not increased, because we wouldn't expect the stoat population to subsequently increase and have a negative impact on Kiwi. It would be more cost effective to wait until rat numbers start tracking up. So this slide explores the question of when to apply 1080 relative to a masting event. So the black line indicates a hypothetical scenario of rat increase over time following a masting event. So time is on this x-axis, and rat abundance will increase and then decrease to very low levels after the mast. So the question is, do we apply 1080 at the first sign of an increase in rat abundance, as indicated by this blue arrow here? Or do we let the rat population grow a little bit and then apply 1080, as shown with this red arrow? And what's going on here, the logic here is that if there are a few more rats out there, there will be more rat vectors to get 1080 into stoats, which is what is the Kiwi chicks are most vulnerable to, are the stoats. So this might increase the impact on stoats. Now, having said that, if the species we were interested in was vulnerable, the bird species we were interested was vulnerable to rats, we definitely would not want to let this rat population grow and apply 1080 here, because those rats, in the meantime, would be depredating the bird species that we were trying to predict. So we'll come back to this issue of how different species will result in different strategic outcomes. So in this slide, over here on the left, you can see a particular management zone, the Mount Forbes management zone in Fiordland, and the red gradient here is relative stoat abundance. So dark red and dark colors are high stoat abundance, and light red colors are low stoat abundance, and there are some areas with no stoats. And if we apply 1080, we see that stoat densities are decreased everywhere, pretty much, and that the surviving stoats are patchily distributed. And that leaves some big gaps in the landscape where stoat density is very low, or stoats might even be absent for a time being. And that time is hopefully long enough, or should be long enough, for a subset of Kiwi chicks to grow to be 1 kilogram or bigger, at which point they would no longer be vulnerable to stoat predation. But of course, this situation won't last forever, because these survivors will reproduce. stoats are highly reproductive and they can disperse long distances. So there will be recolonization into this area. But an important point is that to have significant Kiwi outcomes or improvement in Kiwi populations, we don't have to remove all the stoats. We just have to create these areas where a subset of Kiwi juveniles can grow big enough to escape predation. So to begin to summarize here, we've developed a broad-scale model that is generally adaptable to any species or any ecosystem and any scale that we want to address. And it takes the broad-scale processes of masting, disturbance, and dispersal to link the fine-scale dynamics that are going on in widely separated areas between forest productivity, rats, stoats, and Kiwi. So the outcomes all depend on these dynamics of forest, rats, and stoats. And the best Kiwi strategies that are emerging show that we really need to know what's going on with rats in this environment. And we should be responding to rats, and that's because stoat populations will subsequently increase if rat populations grow too high. So earlier I alluded to different outcomes for different species. So the strategy development is really species specific. In the case of Kiwi, the juveniles are vulnerable to predation by stoats alone. But if we were looking at a species that both adults and juveniles were vulnerable to predation, the outcome would be quite different. Similarly, if rats, stoats, and other predators were the problem, then the outcome would be quite different. Our predicted strategies would be quite different. So the next step is really to be able to identify strategies that maximize outcomes for multiple endangered species. But this is complex. I mean, we can do this from a modeling perspective. It's complex, but we can do it. But I think identifying the objective is going to be the hard part, the management objective, because among that species pool of endangered species, there's going to be winners and losers, depending on when and where we apply our management. So this is an important next step, but it's not for today. We can maybe come back in a year's time and address that. Again, an important motivating factor for this work is really to have a mechanism for aligning management biodiversity objectives with what is feasible, and that really any new or emerging project should begin with a careful analysis of a range of feasible outcomes, and a model such as this one provides the ability to do that. Now, the model's not perfect, and it will improve as our ecological understanding improves, but it will also improve with management applications that find that the model doesn't live up to what it's predicted. And that allows us the opportunity to improve the model, adapt the model, given with learnings as we go. And again, so if our initial objectives turn out to be not feasible in the development of a new project, then those objectives will have to be adjusted, or new financial resources need to be obtained so that management can actually achieve the objectives that we set out for. So thank you very much for your attention.
Kia ora, and thank you very much, Dean. That was really interesting, and I can tell that people are interested because we've got some great questions. I'm going to start with a really short one. Dean, how do you determine stoat abundance? Now that came in when you had the slide up there with all the little squares in that management area. How do you know?
Well, that's based on it's an approximation, really, but it's based on previous studies of stoat abundance in this landscape, really, and those stoat and the dispersal ability and survivorship and fecundity. And so we play those dynamics out over time, and you come to a sort of an equilibrium level of stoats, which varies with rat abundance. And but then the application of 1080 can decrease it. So we're really trying to build on existing studies of stoat abundance.
Great, thank you. Now, Tan had a really good question. Why are you looking at timing of 1080 application after the mast if the mast trigger itself was not found to be that effective in management strategy?
Mast. Yeah, well, that's a good question and maybe confusing the way I presented it. So we can look at masting in conjunction with monitoring rat abundance, or we can look at rat abundance just by itself. Either way, and in either case, as rat numbers are ticking up, we begin to ask the question, well, when do we apply? Do we apply now or do we wait a bit so we can have more 1080 vectors?
That's great, thank you. And we've probably got time for one more. Do you model stoat declines only by secondary poisoning, or do you also include declines due to the reduction in their primary prey, i.e. less rats?
Yes, both, yes. So rats really determine sort of how many stoats can be in a particular area. There's sort of they to throw in a little jargon, they sort of set the density dependence dynamics of that area. And then of course, the 1080 can decrease the stoats.
Okay, and we have got time for just one more. What impact does this have on mice? And are stoats likely just to start eating mice instead of rats?
No, that's an excellent question, and one we've swept under the rug for the time being, because mice are very complicated and our understanding of all those dynamics is a little bit limited. And so as this is a modeling exercise, we try to minimize the complexity, especially when we don't know very much about it. And so we've basically just wrapped up mice into the rats.
Okay, little rats. Right, with that, look, we are bang on 11 o'clock, so we need to finish here. Rest assured, there will be responses to all the questions that everyone has asked, and we really appreciate that interaction with you. And we'll follow up with those. We will be back with further webinar on emerging contaminants. So a new topic completely later in the month, and then more on the Kiwi Rescue Program in July. So with that, I'd like to say thanks and goodbye from us for this time.